This figure summarizes approaches taken to date in pursuit of the goal of engineering HSCs. Strategies can be classified as utilizing either morphogen-directed differentiation or transcription factor-mediated conversion. One example of directed differentiation is the strategy used by Keller's group, where pluripotent cell embryo bodies, either from embryonic stem cells or induced pluripotent cells, are initially exposed to morphogen signals that induce specification of mesoderm, which further differentiate to produce primitive hematopoietic progenitors. Subsequently, exposure to a cocktail of pro-hematopoietic cytokines results in the emergence of hematopoietic cells with definitive characteristics including RUNX1, GATA2, and SOX17 expression, as well as T lymphopoietic potential. Several conversion and respecification strategies have been used. Papers from the Moore and Lecoq groups showed that exogenous hematopoietic transcription factor expression can respecify fibroblast to hematopoietic cells. For example, the Lecoq group expressed a defined set of transcription factors in murine fibroblasts. After 10 to 12 days of culture on stromal cells, an endothelial to hematopoietic transition could be detected, with expression and subsequent downregulation of endothelial genes and acquisition of definitive hematopoietic properties. Our group used a combinatorial approach beginning with exposure of human pluripotent embryoid bodies to a cocktail of pro-hematopoietic cytokines for 14 days. This results in the emergence of a mixture of primitive and definitive progenitors that possess restricted myeloid colony forming potential and which are not engraftable. Transduction of these progenitors with 5 pro-hematopoietic transcription factors subsequently confers definitive characteristics including engraftment in immunodeficient mice. Sandler and colleagues took the innovative approach of transducing human endothelial cells with pro-hematopoietic transcription factors. After culture in an in vitro vascular niche construct for 4 to 6 weeks, definitive HSC gene expression patterns emerge along with definitive hematopoietic characteristics, including serial engraftment in immunocompromised mice. The Moore group built on their prior findings in reprogramming fibroblasts to hematopoietic progenitors by using surface markers from hemogenic endothelial intermediates identified during the reprogramming process to isolate placental hemogenic progenitors which, following in vitro co-culture with cells expressing Notch ligands, gave rise to definitive hematopoietic cells with serial transplantation ability.